So this video is a short how we know kind of video. And so the idea is I'm highlighting a technique that we use to look at different proteins that are binding at promoters of genes. So the technique is called DNA footprinting. So the idea here is we're looking for the presence of a protein. The presence of the protein leaves a footprint behind. And so presence or absence can be detected using this technique, DNA footprinting. So the idea here is we're talking a lot about different transcription factors that are binding at promoters. And so I wanted to share this how we know video to help illuminate the techniques and the tools that help us understand why or how things are binding at promoters. So up here we've got an alternative sigma factor, so sigma factor 38. If you don't know what an alternative sigma factor is, I recommend you go back and watch the transcription initiation video. You should start there. This should be a follow-up to that. So the alternative sigma factor is bound here. The gene that we're interested in is called OSMY. And so what we've done is we've taken this piece of DNA, we've taken it out of a cell, and we've left it so that the protein should still be associated. So we're being very gentle with the detergent that we're using, and we're trying not to denature protein as part of our extraction protocol. Once we get the DNA out, we can use a restriction endonuclease that cuts at a specific sequence in the OSMY gene, and then we can use the sticky end that's left behind in order to add a radiolabel to that piece of DNA. And so this is the promoter region up here. So this would be upstream of the OSMY gene, and then the terminator would be down here somewhere off the end. The terminator has been broken off. And so what we're doing, like I said, is we're bringing the DNA out with the protein still associated. We're radio-labeling so that we can detect the DNA. So we don't need ethidium bromide or anything on the back end of this. We're going to use our radio label in order to find the piece of DNA. We're going to take the DNA and we're going to expose it to DNase. And so DNase 1 is an endonuclease that cuts pretty liberally. So it doesn't use a specific sequence like restriction endonucleases do. It'll go through and it'll cut at random places throughout the genome. So what we're doing is we're hoping that the association of the protein with the DNA is going to prevent the DNase from cutting. And so that'll leave the footprint behind. So over here, we've got samples without protein, so we can be pretty harsh with our detergent. That'll strip all of the proteins away, add the DNase, and it'll cut liberally throughout that piece of DNA. Over here, with the presence of the protein, we've got sigma 38 and we've got RNA polymerase bound. If you add the DNase, it's going to cut only places where the protein is not associated. Okay, so you'll get a break here, you'll get a break here, break here, break here, break here. It's going to be random. It's going to be at different places throughout that piece of DNA. Random is actually good in this case. So we can isolate the DNA away from the protein. So we're denaturing here and we're stripping the protein off. And now we can take these pieces of DNA that have radiolabel on one end and we can run them out on a gel electrophoresis. And so here, in the presence of sigma 38, we get some cuts. We get long fragments here and then short fragments down here. And there is a gap in the middle, or a place where the sigma 38 has protected the DNA from being broken by the DNase 1. If we added sigma 70 instead of sigma 38, sigma 70 wouldn't be expected to bind to the promoter of OSMY. And so even in the presence of the sigma factor, we're not getting a DNA footprint. We're not getting a footprint from that particular piece of protein. And so then this last lane is DNA alone. So in this case, we have stripped away all of the proteins and we haven't allowed any protection. And so using DNA footprinting, we can figure out which proteins are binding to a particular promoter, and we can get some approximation of where they're binding because we're using size exclusion to sort these molecules. Largest molecules will be at the top, smallest molecules will be at the bottom. And so this footprint right here would be from the sigma 38. And you can get a rough approximation as to where that protein is binding. So I've got a question about this. I think it's a pretty interesting technique, and hopefully it adds some depth to our understanding of how we know where sigma 70 is binding, where sigma 38 is binding, and we could use this to think about any kind of transcription factor. We could use DNA footprinting to figure out how any kind of transcription factor is binding to DNA.